Hello everyone, welcome to the Fundamentals of Pharmacogenomics Part 2. In this set of slides, I'm going to review with you the some basic genetic and pharmacogenetic nomenclature. I'm also going to take you back to just a little bit to Biology 101, because I realize if you're like me, it's probably been a long time since you had Biology 101 material. I promise we won't do Punnett squares, we won't be talking about pea pods or anything like that. We'll try to keep it focused on the information that's relevant for a clinician to use in clinical practice. So as I mentioned before, in each slide set I will have learning objectives for those of you that like to review some of the concrete actionable items within a lecture. So there's 2 slides of those. So I'm going to start off, or let's start off talking about kind of the big picture in terms of the fundamentals of genomic organization. And if we kind of look at kind of the biggest picture view, it takes us to the genome, which is an organism's complete set of DNA, including all of its genes, regulatory elements, and other nucleotide sequences. So we can think of a genome like a book. And within that book, we have chapters, or chromosomes. And so chromosomes are single pieces of DNA containing many genes, regulatory elements, and other nucleotide sequences. And we know that we have 46 chromosomes, that we have 2 sets of 23. We get one from our mom and one from our dad. We know that chromosomes are made up of genes, or another way to think about it is that chapters are made up or include sentences. So a gene is a region of DNA that contains the code for a biological component, usually a protein. And that's really the perspective that I'm going to be talking quite a bit about or from during this program. So I'm going to be talking a lot about genes, and we're going to be talking about how they usually code for a protein and how that influences disease or drug response. And we know that genes are made up of nucleotides, which are the building blocks of DNA. So another way to think about this is that the sentence is made up of letters, or the alphabet. So our genes are made up of nucleotides A's, C's, T's, and G's. And so this just gives the overall picture of genomic organization. Now, since we're going to be talking a lot about genes, I want to just do a very, very simplistic overview of gene expression. For those of you who are more basic scientists in the group, this will probably put you to sleep because it's so basic, but I think that's really, I want to make sure we all have a basic understanding of it, and I don't think we need to go into gory detail about it in terms of how we're going to apply this information in subsequent lectures that are more clinically focused. And so this overview of gene expression will start here in the nucleus of the cell, and we know that the gene is located in the nucleus, and we know that a gene is transcribed to pre-mRNA. That pre-mRNA contains exons, the red pieces here. It contains introns, the green pieces here. We know that exons code for amino acids. We know that introns are non-coding regions. And what happens is that these get spliced during mRNA processing, and that leaves us this transcript here, which has no introns in it. We have a 5' cap that is added to the transcript, and we have a 3' poly A or multiple A tail that's added to the 3' end of the transcript. And if you remember all the way back to biochemistry, the reason that the 5' cap and the 3' poly A tail are added are to facilitate stability of the transcript and ultimately export into the cytoplasm. So this mature mRNA right here is exported into the cytoplasm, and here is where translation occurs. So the mature mRNA is translated into protein, and that's what's shown by this strand right here. Now, not depicted on this slide is the fact that protein, it doesn't just end there. We know that proteins can do a lot of different things. They can form secondary and tertiary structures. You can have post-translational modifications on the protein that influence health and disease, and proteins can also form complexes with other proteins. Now let's take a closer look at the structure of a gene, and that's shown in the top here. We know that a gene runs from the 5' to the 3' end. And what we're showing you here is a group, this is showing you really the 5' elements in a gene. Many of these are regulatory elements, which I'll talk about in a little bit. Then we get to the coding region. We have exons, which are in green, and in the blue here we have introns. And then we have a 3' untranslated region. We know that the gene is transcribed to form pre-mRNA. That's what I already kind of mentioned on the previous slide. We know that those introns get spliced out, and this is the final transcript with the 5' cap and the 3' tail that ultimately goes out into the cytoplasm and then gets translated into protein. So I showed on that slide a picture of some of the different elements of the gene, so I'm going to give you some formal definitions of those now. So you'll notice that I showed you the promoter region here, this little brown box. Promoter is a really important region. Again, it's a regulatory region. It's the region of DNA that initiates transcription of a particular gene. And it contains DNA sequences that allow RNA polymerase and other transcription factors or proteins to attach and kind of initiate or repress the transcription of that gene. We also talked about the 5' untranslated region, also called the 5' UTR. This is the region of a gene directly upstream from or before the start codon in the coding region. It's also known as the leader sequence. And it contains regulatory elements which also play a role in gene expression. And so this is kind of showing you the 5' UTR right before the start codon there. Then we have the 3' untranslated region, also known as the 3' UTR. This is the region of the gene directly downstream from or after the stop codon or the termination codon. It's also known as the trailer sequence. And the reason why we care about the 3' untranslated region is that it contains regulatory elements that influence post-transcriptional gene expression, stability, etc. And so the 3' UTR is shown here on this picture. So within a coding region, I mentioned that we have exons. This is a portion of a gene that codes for an amino acid or for amino acids. We know that the exonic sequence is retained in the mature messenger RNA. Within these exons, we have codons. These are just 3 nucleotides that code for an amino acid. We also have introns. These are portions of a gene that do not code for an amino acid. These are intervening sequences that are removed during the mRNA splicing process. And we know that genetic variants occur more commonly in non-coding regions, such as introns, than coding regions. And if you think about for a minute why that might be. Well, it's because our coding regions are usually the most important for the function of a gene, and changes in them can be detrimental and have oftentimes bad consequences. So through heredity, coding region sequences tend to be conserved over time. Now that doesn't mean that you can't have genetic variants within an exon. We certainly do have genetic variation within our exonic regions. However, it's just more common if you're comparing where the frequency of variance occurs more often, most likely in non-coding regions. And then we have the intergenic regions. These are the stretches of DNA sequences located between genes. Now, historically, they were thought to be junk DNA. They were thought to have no function. But this idea has pretty much changed over the last, I would say, 10 years or so. And we know now that these intergenic regions are actually quite important because they contain regulatory elements such as promoters, enhancers, and non-coding RNAs. So we're talking a lot about genes. We say when we think about the concept of a gene, it's like a sentence within a chapter within a book. And we know that the gene, the sentence, is composed of the alphabet or the nucleotides, the A's, C's, T's, and G's. So that's adenine, cytosine, guanine, and thymine. And we know that the entire human genome contains roughly 3 billion DNA nucleotides. We often refer to them also as base pairs. And we know that we have about 20 to 30,000 genes in our human genome, although these estimates can vary. The genomes of two unrelated people differ at about 1 in every 500 to 1,000 bases or so. So if you look to your spouse or your friend, your DNA sequences are actually 99.9% similar to each other. And so there's really only a 0.1% difference in DNA between individuals. It's actually a pretty big deal because it gives rise to over 1 million genetic differences between people. And back in 2000, this revelation of human genetic variation, or this breakthrough, got a lot of press and science, and it really was the realization that DNA differs from person to person much more than originally anticipated. So we have many variants or differences in our genome, and we call those genetic variants. These are differences in the DNA sequence compared with a reference sequence. So we have over 10 million variants in our genome. Now, this term genetic variant, we can also kind of subcategorize it a little bit further. We can refer to it as a polymorphism. This is a genetic variant that is common, often defined as 1% or more in the population. Or we can refer to it as a mutation. That's a genetic variant that is rare, often defined as less than 1% in the population. So both polymorphism and mutation, both of those terms fall under the umbrella of genetic variant. The major difference between the two terms is mainly the frequency at which they occur in a population. So if it's common, you call it a polymorphism. Generally, if it's rare, it's referred to as a mutation. Now, there are many different types of genetic variants, like actual kind of differences in or changes in the DNA code. And we're going to review 4 of the major types. We're going to talk about single nucleotide variants or polymorphisms, insertion or deletion variants, variants that are referred to as a variable number of tandem repeats, and then also copy number variants. So let's start off with single nucleotide polymorphisms. It's abbreviated SNP and it's pronounced SNP. A lot of folks now refer to it also as just a single nucleotide variant, or SNV. You can use SNP or SNV, either one. And really it's quite simple. What is it? Well, the difference, or the name, what it is actually in the title of its name. It's just a difference in one nucleotide, one base pair. So at a particular spot in DNA, I might have a C, whereas my friend might have a T. That C-T difference is a single nucleotide variant. Now, if it's common in the population, it's called a single nucleotide polymorphism. So single nucleotide variants account for about 90% of variation in the human genome. We also have insertion or deletion variants, or indels for short. These are an insertion or deletion of multiple consecutive nucleotides in the DNA sequence. So at a particular spot in DNA, I might have 30 nucleotides in a row, whereas my friend might have those 30 nucleotides deleted at that position. And that would be called an indel. Now, you can have one base inserted or deleted. You can have 1,000 bases inserted or deleted. You can have different sizes of indels. So this picture here just is going to illustrate the first type of variant I talked about, the single nucleotide variant. And what I'm showing you here is a coding region of a gene. And the way you know that is that you have the 3 nucleotides, GCA, which code for an amino acid. In this case, it's alanine. So if we look at gene A from person 1 and gene A from person 2, let's compare the sequences at this first codon. So GCA codes for alanine, GCG also codes for alanine. This A-G difference is a single nucleotide variant. But notice it results in the code for the same amino acid. So it's called a synonymous single nucleotide variant, meaning it codes for the same amino acid. And that's possible because we have a degenerate amino acid code. We can have different codons that specify or code for the same amino acid. If I move to the second codon, in person 1, it's AGA. Let's move down to person 3, it's AAA. This GA difference, AGA codes for arginine and AAA codes for lysine. So this GA difference, again, is a single nucleotide variant. It's a difference in a single nucleotide. But notice here that it results in the code for a different amino acid. And in this case, we have arginine versus lysine. So this is called a non-synonymous, or not the same, single nucleotide polymorphism. So I usually ask when I teach the pharmacy students, I said, if I gave you money and I said you can pick this variant in codon 1 versus this variant in codon 2 to study in a pharmacogenetic study, which one would you pick? And most folks say, well, I would pick the second one because it results in the change in the code for an amino acid, and that would be more likely to affect potentially the function of a protein, which might alter something related potentially to drug disposition and drug response. That's not to say that synonymous nucleotide, single nucleotide polymorphisms can't have functional significance, but it's less likely than the non-synonymous single nucleotide polymorphisms. So let's move on to the next type of genetic variant. This is a variable number of tandem repeats, or VNTR for short. And this is a sequence of nucleotides that's repeated a variable number of times. So if you look at this first example here, you see the letters TG, 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 etc. So that TG is repeated 10 times. If you look at the second example, you have CAG, CAG, CAG. We see that CAG is repeated 4 times. And if we go to the third example, we have ATTCG, ATTCG, etc. We see that ATTCG is repeated 3 times. So if you have a repeat that's less than 5 base pairs in length, they're called microsatellites, short tandem repeats, or simple sequence repeats. And so that, for example, would be example A here and example B, because we have, this is 2 and this is less, and this is 3 base pairs in length. Repeats greater than or equal to 5 base pairs in length are called mini-satellites, and that's the example that you have in this last bullet here. So let's talk about some clinical examples of VNTRs. I think a lot of times people are like, well, that's interesting, but how is that clinically relevant? Well, it's actually very relevant for health, disease, predisposition, and also drug response. And I've given a couple of examples here. The first is a disease example. So Huntington's disease, which is a genetic disorder. It's a neurodegenerative disorder that affects muscle coordination, and it leads to cognitive problems and behavioral symptoms as well. We know that it's caused by an increased number of CAG repeats in the Huntington gene. So repeats of greater than 40 are associated with disease expression, whereas repeats of less than 26 are considered normal. And so this is genotyping for this VNTR is one of the ways that Huntington's is diagnosed. Next, we have Gilbert syndrome, and this is a genetic disorder that's caused by a variable number of TA repeats in the UGT1A1 gene. Now, the pharmacists in the group will recognize UGT1A1 as a phase 2 drug metabolizing enzyme. We know that it's very important for glucuronidating bilirubin. We also know it's very important for metabolizing or glucuronidating certain drugs. For example, irinotecan is one. Atazanavir is another one as well. So if we have 7 TA repeats, so TA is repeated 7 times instead of 6 TA repeats, it causes reduced UGT1A1 expression and decreased ability to conjugate bilirubin. And so the hallmark of Gilbert syndrome is actually hyperbilirubinemia. So if some of you have gone to the doctor and you got a basic metabolic panel or comprehensive metabolic panel drawn, and your doctor says, you know, your bilirubin's a little high, but don't worry about it, it's probably nothing. It might be just because you carry 7 TA repeats instead of 6, and that you have Gilbert syndrome. And it's actually quite common in the Caucasian population. So that's just 2 clinical examples of VNTRs. Lastly are copy number variations. And this type of genetic variant has gained a lot of attention in recent years. It's been found that copy number variations, or CNVs, play a really important role in disease and pharmacogenetics. And basically what it refers to is the gain, the duplication, or the loss, the deletion of large regions of DNA sequence. And so this is showing a duplication. It's showing the gene before duplication, and then it's showing the region that got multiplied. And then this example is showing a deletion. And this is showing the region of the gene that's ultimately deleted from the sequence. And so in terms of pharmacy, one of the best examples we have of a gene that has some pretty significant copy number variations is the cytochrome P450, the CYP2D6 phase 1 drug metabolizing enzyme. There's some pretty prominent copy number variations in it. There are duplications, and these can cause a person to be phenotypically what's called an ultra-rapid metabolizer for CYP2D6. There's also deletions of portions of 2D6 or the whole gene, which give rise to poor metabolizer status or phenotypic activity. And we're going to go over all of this in great detail in the clinical pharmacology lecture, but I just wanted to start alluding to some of these terms and how they relate to pharmacy and pharmacogenetics a little bit earlier. There are also a number of other types of genetic variants, though they're listed and defined here just for your information. I tend not to use these terminologies when I teach these topics, but I do want you to have them just for completeness sake. Now, when we talk about genetics, we'll often use the term allele. So let's define what that is. That's one of 2 or more forms of a DNA sequence that's located at a specific position on a chromosome. So we know that humans are diploid organisms. We have 2 copies of every chromosome and gene. So for every position in human DNA, we have 2 alleles. You get one from your mom and one from your dad. The wild type allele is the most common allele, or sometimes it's referred to as the reference or the major allele. The other allele is the other type of allele you can have is a variant allele. That's a polymorphic allele, also known as the minor allele. So the combination of alleles a person carries at a particular spot or location in DNA is called a genotype. Again, remember that one allele is from your biological mother and one allele is from your biological father. So if you have 2 identical alleles, you're considered to be homozygous at that position. If you have 2 different alleles, you're considered to be heterozygous at that position. So in this example here, this first gene is for the copy that you get from mom. The second is the copy that you get from dad. And let's focus on this third position here. Sorry, my arrows are a little bit off. They should be focused on this A and this G. So at this particular spot in DNA, everyone has 2 alleles, right? And this person has an A and a G. So their genotype is AG, because they get the A from mom and the G from dad. Now, I'm going to give you another example to try to get at the terms wild type and minor allele. So the example here is at nucleotide position 681 in the cytochrome P450, the CYP2C19 metabolizing enzyme gene, there are 2 possible alleles, G and A. The frequency of the G allele is 90%, and the frequency of the A allele is 10%. So what is the wild type allele? Well, we know that the wild type allele is the most common or the reference allele in the population. And so in this example, the most common allele is G, because it's in 90% of the population. So what is the minor allele? The minor allele is the other allele, A. So what are the three possible genotypes? So in a population, we know that these two alleles exist at this particular spot or position in DNA. So what are the three possible genotypes that a person could potentially have? Well, it could be GG, GA, or AA. If they're GG, we would say that they're homozygous wild type. GA, we'll say they're heterozygous. And AA, we would say they're homozygous for the variant allele or the polymorphic allele. Now, when we talk about alleles and genotypes, we know that allele and genotype frequencies in a population will remain constant from generation to generation in the absence of other evolutionary influences. And this really is the basis of what's called the Hardy-Weinberg equilibrium principle. So allele and genotype frequencies in a population remain constant over time in the absence of other evolutionary influences. And we know that if we have two alleles, so let's say P and Q are the frequencies of two alleles such that P plus Q equals 1. So P is the frequency of the major allele, Q is the frequency of the minor allele. So two alleles, the frequencies of which are represented by P and Q, and we know that P plus Q equals 1. So if I told you that the frequency of the major allele, or that P, was 60%, you can calculate the frequency of the minor allele. It's just 1 minus 0.6, which is 0.4 or 40%. So pretty easy, and I promise these are probably the only equations you'll see in this entire certificate program. And some of you are like, no, not equations. But I think it's a really important concept just to kind of understand where or how allele and genotype frequencies kind of evolve over time. So we know that we're diploid individuals. So if we take this equation and we square it and then we factor it out, we get P squared plus 2PQ plus Q squared equals 1. So P squared represents the frequency of those who are homozygous for the major allele. 2 times P times Q represents the frequency of heterozygotes in the population, and Q squared is the frequency of individuals in the population who would be homozygous for the minor allele. So this equation is known as the Hardy-Weinberg equilibrium or Hardy-Weinberg equation, and it is able to tell us the expected genotype frequencies in a population, assuming that they remain constant from generation to generation in the absence of other evolutionary influences. So again, this is what you would expect to see in the population, or the frequencies that you would expect to see in the population for those particular genotypes. So what does it mean when the observed number of genotypes in a population deviates significantly from that expected by Hardy-Weinberg equilibrium? Well, it could mean that mating in the population is not random. For example, maybe inbreeding has occurred. It could mean that the population size is too small, or even perhaps that genotyping error occurred in the laboratory. Now, I recognize that many of you are not going to be in the lab kind of doing this work, and you might be thinking, well, how is this Hardy-Weinberg thing relevant to me? Well, it's actually pretty relevant when you read pharmacogenomic or even genomic studies. They oftentimes have to tell you whether the frequencies of the genotypes were in Hardy-Weinberg equilibrium. And if they say they're not in Hardy-Weinberg equilibrium, that should make you question these things here. So that's why I think Hardy-Weinberg is important to acknowledge in this program. So we've been talking a lot about genetics or genotype. Now let's move on to phenotype. So phenotype is the measurable characteristic of an organism. And if you go back to Biology 101, we know that the typical phenotypes that people talk about are hair color or eye color. We know that phenotypes are a result of genetics, environment, or the combination of both. So we know that genetics does play a role in underlying most phenotypes. It's not always the primary driver. You can have environmental factors as well. And in this program, we're not going to be talking in this course, we're not going to be talking a lot about hair color or eye color. We're going to be talking more about pharmacologic phenotypes. So we're going to be talking about metabolism phenotypes, for example, such as ultra-rapid metabolism, extensive metabolism, intermediate or poor metabolism. We might be talking about pharmacologic phenotypes such as plasma drug concentrations and how they are similar or different between people with different genetic makeups. There's some other PK parameters as well. Or we might be talking about pharmacologic phenotypes from the pharmacodynamic perspective, such as someone who's a responder to a drug or a non-responder to a drug. So let's delve a little bit deeper and talk more about the different types of genetic variants and how they're functionally significant and potentially could give rise to different phenotypes. So first off, I think really important is that genetic variants can occur anywhere in a gene. They can occur in the promoter, an exon, an intron exon splice junction, the 3' untranslated region. And oftentimes the type of variant and its location in DNA influence its function. So this slide reviews the potential functional consequences of variants in different regions of a gene. And I refer you to that pharmacogenomics primer I posted. I think this primer, it's almost 10 years old, or actually is 10 years old now, but I think does a really good job at illustrating, showing pictures of how variants in different regions of the gene ultimately can affect function. And so I'm not going to walk through all of the pictures in that primer. I'm going to walk through one example, but I do refer you to that primer for pictures of, I think, all of these examples. So if you have a variant in the promoter or 5' untranslated region, we know that that may alter transcription factor binding and increase or decrease gene transcription. So we know that transcription factors recognize specific sequences in the promoter in the 5' untranslated region. And when they recognize them, they can turn on or repress gene transcription. And so if you have a variant which alters that sequence, that can alter how that transcription factor binds and ultimately how it governs gene transcription. Variants in an exon may change a codon sequence, and we looked at this kind of example in multiple slides so far. So it can change a codon sequence, resulting in a different amino acid, or maybe even a premature stop codon, or it may result in the code for the same amino acid. Variants in an intron, they're very common, as we mentioned, but they often have no functional consequences. And why? Well, because they're spliced out during mRNA processing. So some of you might be thinking, well, I saw this paper that was published where a variant in an intron was associated with response to, I'm just make up a drug, let's say acetaminophen, for example. So is that paper bogus? Well, no, not necessarily. An intronic polymorphism or variant may have consequences if it's quote-unquote linked to other variants in the gene. And I'm going to talk a little more about linkage in a few slides, so I think this will make more sense. Variants can also occur in the intron-exon splice junction, and that may alter the splicing of the transcript. So in those intron-exon splice junctions, you have specific sequences that a spliceosome recognizes. If that sequence is altered because of a genetic variant, it might alter how the transcript is spliced, and that might result in a transcript that lacks exons or maybe even contains pieces of introns. And but we have some really good examples in pharmacogenomics of how these kind of splicing variants can greatly impact drug response, and we'll talk about them later on in the course. And lastly, variants in the 3' untranslated region. They potentially can alter mRNA stability, structure, or degradation. So as I mentioned, I'm going to just show one picture from that primer, and this is an example of genetic variants in the protein coding region, or the exon. And this again is just reviewing kind of what we reviewed before, but just showing it in a different picture form. So this is showing you the reference DNA sequence, and we'll focus on this spot here. CGA codes for arginine. And if we move down to the second copy here, we see AGA. This CA difference between people is a single nucleotide variant, but you'll notice that it results in the code for the same amino acid. So again, that's called a synonymous single nucleotide polymorphism. And again, those often have no functional consequences. There are a few examples in the literature where synonymous SNPs have been linked to functional pharmacologic phenotypes, but really they're few and far between. And then if we look at this first example, CGA codes for arginine, and then GGA codes for glycine. Again, this CG difference is a single nucleotide variant, and notice that it results in the code for a different amino acid. Again, that's a non-synonymous polymorphism. And these are ones that tend to be studied quite often in pharmacogenomics. They can result in increase or decreases in protein function or quantity. They may result in a premature stop codon, resulting in a truncated protein, which may be inactive or degraded. And I actually deleted the next slide so you can cross that out. Or they potentially could have no functional consequences. So let's switch gears. So we've talked about different types of genetic variants. We've talked about their functional consequences. Now let's switch gears and talk a little bit more about the nuts and bolts of genome logistics, if you will. So the first major logistic is, well, where does the human genome live? So if you're bored on a Friday night, you don't have anything to do, you've watched all possible episodes of Alaskan bush people like I have, you can go on the NCBI website and kind of pour through the human genome. A really exciting activity. So this is where the human genome is housed. Now, how is the human genome kind of referred to in these databases and also in publications? Well, generally the reference sequence, also called the RefSeq, is the basic level of sequence that is used. So it could be the genomic DNA level, the coding DNA level, the protein level. And this will make sense, and hopefully when I get to the bottom of the slide. So most of the time you're going to see the genetic nomenclature presented at the DNA level, where nucleotides are designated by the bases A, C, Gs, and Ts. Other times you may see the information presented at the protein level, where it's represented by one-letter amino acid codes, like we've already talked about. For example, arginine, I think K is lysine, and V for valine. You might see an X, which designates a translation termination codon. Genetic variants are designated by this greater than or this caret character. And so if there is a single nucleotide variant in the population, let's say a G to T, you'll see G caret T. Now, to avoid confusion, the way that the nomenclature, the way that you present the information, should be preceded by a letter indicating the type of reference sequence that is used. So G dot for a genomic sequence, C dot if you're presenting the coding DNA, or P dot if you're presenting a protein sequence. And I have a couple of examples of this, so I think it'll make more sense. So let's look at this, this information here. So it's CYP2B6, C dot 516, G caret T. So the first few letters, numbers here in orange, identify the gene name. And in this case, the gene is cytochrome P450 2B6, which is a phase 1 drug metabolizing enzyme. Now, gene names are typically italicized. So if we're talking about the CYP2B6 gene, this CYP2B6 would be italicized. If we're talking about the CYP2B6 protein, we would not italicize the word CYP2B6. The numbers following the gene name indicate the nucleotide position in the gene. So this is the yellow part here. So 516 is the nucleotide position in the gene. The lowercase c tells you that the reference sequence that is being presented or that's being used is the coding sequence. So in the coding region of the CYP2B6 gene at nucleotide position 516. The first letter represents the wild type or reference allele, so in this case it's a G, and the second letter represents the variant or polymorphic or the minor allele, in this case it's T. So G is the wild type, T is the variant. So let's look at this example. We have CYP2B6 P.Q172H. So again, the first few letters or numbers identify the gene name. The numbers following the gene name indicate the codon position in the gene. So you have P.172. So how do I know that it's the codon position? Well, the lowercase p indicates that the reference sequence that's being presented to me is a protein sequence, and we know that protein is presented in the form of the amino acids, and therefore we have to think about the codon number of that amino acid. The first letter here represents the wild type or reference amino acid, Q, which is glutamine. The second letter, H, represents the variant amino acid, which is H, or histidine. So if you take 172, I'll review this again. So in the CYP2B6 gene, we're talking about it here at the protein level. So at codon 172 in the CYP2B6 gene, there are two possible alleles or amino acids, if you will, Q or H. And if you take 172, the codon, and you multiply by 3, guess what you get? You get 516. So what this is showing is that you can refer to the same variant a number of different ways. So you can refer to it at the coding sequence level, the kind of DNA, the nucleotide level, such as this, or you can refer to it at the amino acid or the codon level, such as this. So both of these examples are the same variant. It's just depicting them using a different type of kind of reference nomenclature. So I have some practice examples at the end of the lecture that I'm not going to go over with you. I'll post the answers to them next week, but it allows you to kind of practice more with the nomenclature. It starts to make more sense the more you kind of work with it and practice it. So how do we actually catalog all these genetic variants? You know, I mentioned that there's millions of variants in our genome. Well, they're actually very well categorized in a database called DB-SNP, or Database for Short Genetic Variations. Again, when you're bored on a Friday night, you can go onto this website and plug in your favorite single nucleotide or whatever kind of variant that you like to research. All validated variations in this database are assigned what's called a reference SNP number, or an RS number, for identification and retrieval purposes. For example, this variant that I just showed you, the 516 variant in the CYP2B6 gene, the G to T variant, it is tagged as RS3745274. So if you plug this into the DB-SNP website, it will pull up this variant for you, and it will tell you a number of things about it, like the frequency of the alleles in the population, in different populations, the frequency of the genotypes in the different populations. It'll tell you about basically that it results in a change in the code for an amino acid at codon 172. It'll show you this information here, and it'll show you actually a snippet of the sequence of kind of where it is located. So it's actually a really great database that has evolved over the last, I'd say, 10 years or so. Back when I was in training, we used to, God, I sound really old right now, but we used to actually have to look up a lot of genetic sequences in kind of the primary literature, and you'd be mapping them by hand. And now all you have to do is plug in the RS number, which is usually widely reported and publicized in the literature, and voila, all the information is right there for you. dbSNP, as I mentioned, also provides information on the type of variant, the sequence, genotype, and allele frequencies in different race and ethnic groups. Now, lastly, I want to switch gears. We've been talking a lot about genotypes, basically what you get from mom and the allele you get from mom and the allele you get from dad. I want to switch gears and instead not focus on the two chromosomes, but just focus on a single chromosome, or a haplotype. So a haplotype is a set of closely linked alleles that are located on one chromosome and inherited together as a unit or a block. So when we talk about genotype, we're talking about one allele and two chromosomes. When we talk about haplotype, we're talking about multiple alleles on one chromosome. So I'll let that sink in for a minute. And these alleles tend to be inherited together as a unit or a block. Haplotype blocks may contain a large number of variants, but typically a few SNPs in the block are enough to uniquely identify the haplotype in that block. And so we often have our what's called tag SNPs. These are a SNP that is representative, it's the representative SNP in a region of the genome with high linkage that represents a group of SNPs in a haplotype block. So I'll talk a little bit more about this in a minute, but I first want to get to this word I just used, linkage. And what that's getting at is the term linkage disequilibrium. And that's the non-random association of alleles at different loci on the same chromosome. So it's the phenomenon by which the presence of one allele in a chromosome suggests a high probability that a particular allele will be present at a neighboring site on the chromosome. The degree of linkage between two alleles is scored from 0, which is no linkage, to 1, which means two alleles are always linked in a given population. So if you think about it, if we were all genetically identical and our genome never changed, the linkage, the LD score, between two alleles would always be 1. Now sometimes, for certain alleles, that is the case, but because we are all different and our genome does change, it's not always 1. And so, again, I just want to reiterate this haplotype concept, or this haplotype term. So again, during recombination, chromosomes containing segments of DNA are taken randomly from the two different parental chromosomes. And generally, sequences of DNA that are close together tend to be inherited together in units or blocks. So again, a haplotype is a set of closely linked alleles that are located on one chromosome and inherited together as a unit or a block. So this is showing you a depiction of the haplotype. So again, we're showing one chromosome here, and we're showing 3 different SNPs. We have a TC at this first position, an AG at the second position, or a GT. So the 3 different potential haplotypes that a person could inherit might be haplotype 1 down here, such as T, A, G. They could potentially inherit haplotype 2, which would be C, G, G, or haplotype 3, which would be C, G, T. So those are, again, the linear arrangement of alleles located that tend to be inherited together and located on one chromosome. Again, they tend to be inherited as a unit or a block. So I'm going to complicate things a little bit further. This is the part that usually drives people crazy. And I'm going to bring up the term diplotype. This is the specific combination of 2 haplotypes. So what an allele is to genotype, haplotype is to diplotype, or vice versa. So what genotype is to allele, diplotype is to haplotype. So if we take the example here, you have mom and you have dad, and mom has the, let's say, the *1 haplotype and the *2 haplotype. Her diplotype is *1, *2. So we know that she has this the star 1 haplotype, which is, as we already talked about, the TAG, and she also has the star 2, which is the CGG. Dad has 2 copies of the star 3 haplotype. So Dad has 2 copies of this haplotype here. And so what do they pass along to their offspring? So let's say their son inherits a star 2 from mom and a star 3 from dad, and their daughter inherits a star 1 from mom and a star 3 from dad. And so again, the diplotype here would be star 2, star 3, or star 1, star 3. So basically, the way I like to think about diplotype is that it's the genotype of the haplotype. I don't know if that's helpful or more confusing, but it's basically just the specific combination of 2 haplotypes that a person has. So a really important project has been completed called the HapMap project. And the purpose of the HapMap project was to provide a map of those haplotype blocks and the specific tag SNPs that identify those blocks for the entire genome in different populations around the world. So you might be thinking, why does Chris keep talking about haplotypes and tag SNPs? Why are these important? Well, by identifying regions that are inherited together and then being able to tag them, the HapMap project basically reduces the number of variants that are required to examine the entire genome. So if you have 10 million, let's say, single nucleotide variants in the genome, we know that some of those are inherited together. And so if we can figure out which ones kind of tag a particular region or haplotype, we can dramatically cut down the number of variants we have to interrogate in the genome to provide us information about a larger percent of the genome. So why is this important? Again, it improves the efficiency of genomic studies. Effort is not wasted on genotyping redundant variants. And this has been taken even a step further with the 1000 Genomes project. And the purpose of this project is to provide genotype and haplotype information on all types of genetic variation in 2,500 people from 26 populations around the world through the use of whole genome sequencing. And the goal here is to provide a deep catalog of human genetic variation and to identify greater than 95% of genetic variants that have frequencies of greater than 1% in the population studied. So instead of just looking at the typical populations that, or the first populations that the HapMap project looked at, for example, like a Caucasian population or an African population, this has expanded the genetic interrogation into many different populations from around the world. And this is important because we know that genetic architecture is not the same between all races and ethnicities. And so you really do have to finely characterize or categorize variants in a specific population to have a good assessment of what's going on with that particular population's genetic architecture. Now that wraps up this presentation. After this slide, you'll see some additional practice slides for your enjoyment. I will post the answers to those practices next week. And just want to reiterate, as I said at the beginning of this course, the purpose, one of the benefits of this is that you have access to me throughout the entire course. So please don't hesitate to contact me if you have any questions. Thanks so much.